Welcome to today's lecture on cell biology. Today, we will be exploring the fascinating world of cell signaling. As you know, cells communicate with one another through a variety of molecular mechanisms, allowing them to coordinate their functions and respond to changes in their environment. This intricate process of signaling is essential for the development, maintenance, and regulation of various physiological processes in the human body. Paracrine signaling is a common type of cell communication where cells release ligands that diffuse to nearby cells. Ligands can be proteins, like growth factors, or small molecules. Ligands bind to receptors on target cells, activating signaling pathways that can change gene expression, cell behavior, and function. Autocrine signaling is a self-stimulatory process in which cells release signaling molecules that bind to their own receptors. This type of signaling is important in maintaining cellular homeostasis and regulating cell growth and differentiation. In autocrine signaling, cells produce and secrete ligands that bind to receptors on their own surface, initiating intracellular signaling cascades. This can lead to cellular responses such as survival, proliferation, or migration. Synaptic transmission is a specialized form of cell signaling that occurs between neurons. In this process, a presynaptic neuron releases neurotransmitters into the synaptic cleft, which then bind to receptors on the postsynaptic neuron. This interaction between the neurotransmitters and receptors triggers a series of events that allow for the transmission of electrical signals from one neuron to another. This communication is vital for processes such as memory formation, motor control, and sensory perception. Hormonal signaling involves the secretion of hormones by specialized cells or glands into the bloodstream. These hormones travel throughout the body and bind to specific receptors located on target cells, initiating signaling cascades. Hormonal signaling plays a crucial role in regulating various physiological processes, including metabolism, growth and development, reproduction, and stress response. Examples of hormones include insulin, estrogen, testosterone, and adrenaline. Apoptosis, or programmed cell death, is a tightly regulated process crucial for maintaining tissue homeostasis and eliminating damaged or unwanted cells. Signaling pathways involved in apoptosis play a significant role in development, immune response, and prevention of cancer. Multiple signaling pathways, such as FOS/fossil, TNF-alpha, and p53, are involved in apoptosis. These pathways activate specific proteins that ultimately lead to the dismantling of cellular components and the removal of the cell without triggering an inflammatory response. Notch signaling is an evolutionarily conserved pathway that regulates cell fate determination, tissue development, and homeostasis. It plays a pivotal role in processes such as embryonic development, stem cell maintenance, and immune system regulation. The Notch pathway involves the binding of Notch receptors on one cell to ligands on a neighboring cell. This interaction triggers proteolytic cleavage, releasing the Notch intracellular domain, NICD. NIC then translocates to the nucleus, where it acts as a transcription factor, regulating gene expression and influencing cell fate. JAK-STAT signaling is a major pathway involved in transmitting signals from a variety of cytokines and growth factors. This pathway is essential for regulating immune responses, cell proliferation, and differentiation. In JAK-STAT signaling, cytokines bind to specific receptors, leading to the activation of Janus kinases, JAKs. Activated JAKs phosphorylate the receptors, providing docking sites for signal transducers and activators of transcription, STATs. The phosphorylated STATs translocate into the nucleus and regulate the transcription of target genes. The WNT signaling pathway is critical for embryonic development, tissue homeostasis, and stem cell renewal. Dysregulation of this pathway is associated with various developmental disorders and cancer. WNT signaling involves the binding of WNT ligands to frizzled receptors, leading to the activation of downstream signaling cascades. These cascades regulate the levels of beta-catenin, a key component of the pathway, which then translocates to the nucleus and modulates the expression of target genes. Cell adhesion molecules, CAMs, are proteins present on the cell surface that mediate cell-cell and cell extracellular matrix interactions. These interactions are vital for tissue formation, immune response, and cell migration. CAMs play a crucial role in cellular processes such as embryogenesis, wound healing, and immune cell recognition. Examples of CAMs include integrins, cadherins, and selectins, each serving a unique function in cellular adhesion and communication. Intercellular communication channels, such as gap junctions and plasmosmata, facilitate direct communication between adjacent cells. These channels allow for the exchange of ions, small molecules, and even organelles, enabling coordinated cellular responses. Gap junctions, found in animal cells, consist of connection proteins that form intercellular channels. Plasmosmata, on the other hand, are plant-specific channels formed by the ER membrane. These channels establish cytoplasmic continuity between adjacent cells, promoting signaling and nutrient transport. Quorum sensing is a communication mechanism used by bacteria to coordinate group behavior based on population density. It allows bacteria to synchronize gene expression and collectively respond to changes in the environment. In quorum sensing, bacteria secrete signaling molecules called autoinducers. As the population density increases, the concentration of autoinducers also increases. Once a threshold concentration is reached, the autoinducers bind to receptors, triggering specific gene expression patterns and physiological responses. The immune system relies on intricate signaling networks to coordinate the response to pathogens and maintain immune homeostasis. Immune cell signaling involves the activation of immune cells, such as T cells, B cells, and macrophages, in response to the presence of antigens. During immune cell signaling, various receptors on immune cells recognize specific antigens, leading to the activation of downstream signaling events. These events can involve cytokine release, antibody production, and cellular immune responses, ensuring an effective defense against pathogens. Cell signaling plays a vital role in coordinating and regulating physiological processes. From paracrine to immune cell signaling, we have seen the diverse mechanisms employed by cells. Understanding these intricacies improves our comprehension of cell function and aids in targeted therapeutics and disease treatments. Let's continue exploring this captivating field for future medical advancements.